This video is about DNA replication, or how DNA is copied from parent to offspring. This is part of a video series on DNA in general and gene expression. Why would a cell want to replicate its DNA? To make new cells. Up here in the top left corner, you can see one bacteria copying its genome. 1, 2, now it's got 2 copies, and then the cell dividing around the 2 copies of the genome into 2 identical daughter cells, where once you had 1 bacteria, now you have 2. The younger cells we call daughter cells, regardless of the actual sex of the organism. So you have parent cells always making daughter cells. So when single-celled organisms like bacteria form daughter cells, those are the new individuals in the bacterial colony. But when you or a plant or a fungus make new cells, it's either to make sex cells for reproduction or to grow your body. Your body is like a colony of cells, except with strong attachments holding them together. So all cells have daughters by copying their DNA and then dividing in half. This results in cells doubling in number. So it happens here. You have one single egg. This is just one cell, and it got fertilized by a rooster sperm in this case, and became a fertilized egg. And inside you see this embryo growing. Well, that's because the one fertilized egg cell, which was very tiny inside the egg, it was nourished by the yolk and the egg white, and it was just microscopic, and it just grew and grew, and it grew in number. It grew and divided, grew and divided, grew and divided. Very similar, actually, to what this bacteria was doing over there, until it became this big mass of cells that we call an embryo. And the embryo grew into a hatchling, and the hatchling grew into a chick, which grew into a chicken. These organisms, each stage of this chicken cycle, the organism, the living organism, is bigger and bigger and bigger. And it gets bigger because you're just adding numbers of cells. And that happened by the original fertilized egg copying its DNA and then dividing in half, just like we saw with the bacteria. Same thing happens with fungi. They start as little spores, and then the spores grow and divide, grow and divide, grow and divide, until they create this large, complicated organism, a mushroom. Same thing with seeds. Plants do have sperm. The sperm is contained inside the pollen, and it's received by female flowers, which have eggs inside of them. And so when you get a seed, that's going to be a fertilized egg. A plant egg and a plant sperm. Plant sperm are not swimming, but just to make a symbol that you recognize, there's a swimming sperm. They came together to form a seed, and then the seed grew and divided, grew and divided, grew and divided, until it could create new plant parts. You get a germinating seedling, and then a mature plant. So every daughter cell inherits DNA from its parent cell. The parent's DNA has to be copied before the cell divides so that the daughter cells can both inherit the DNA of the parent. So when do cells copy DNA to daughter cells? Well, they could do it for reproduction, and that can be either clonal reproduction, like we saw with the bacteria. That means no sex required. The parent cell is just making a perfect copy of itself when it creates its daughter cells. Or sexual reproduction. Production of sperm or egg also requires DNA to be copied to daughter cells. Also for growth, for an organism to grow bigger, a multicellular organism to get bigger, we just need to add cells. So the cells need to copy themselves. Healing, wound repair. If you get a wound, you've damaged or lost certain cells. So this is another time where parent cells that are already there need to create daughter cells to replace the cells that were lost. Replacing cells that have reached the end of their lifespan. Cells have a lifespan as well, so they'll age out, and they have to create daughter cells that can continue on after they are gone. So in these pictures, I'm just reiterating what we've already talked about. You start with a single fertilized egg. That's a human zygote. There's already been a sperm absorbed into it. And once the sperm has fertilized the egg, that egg begins to divide until you have a mass of cells called an embryo, which continue to grow and divide until they grow into a fetus, which grows into a baby, and so on and so forth. So DNA replication follows what's called a semi-conservative model of replication. So first, the two parent strands of DNA separate. So here you have the parent strand. The parent strand is always going to be blue. And you see that it separates. So some enzyme comes along and separates the two strands and adds complementary nucleotides to form two daughter strands. These are daughters. Now, these nucleotides are added following the complementary base pairing rules. So wherever there was an A in the parent strand, we add a T, because A's and T's always bind together. Wherever there was a C in the parent strand, for the daughter strand, we add a G, because C's and G's bind. G's to C's. Here, we're going to add a T, and to this T, we will add an A. Now, these nucleotides were just free-floating in the cell. You got them from your diet. You ate some animals, you ate some plants, and extracted their DNA, broke it into bits, and now you're using it to those nucleotides from your diet to build new DNA for your cells. So the two parent strands separate, and nucleotides are added following the complementary base pairing rules to create the two daughter strands. This is called the semi-conservative model because half of the parent strand, the older parent strand, is conserved in each new daughter strand. Now, these are each going to go to a daughter cell. We probably can't see what I'm writing because my image is probably there, so let's write it over here. One daughter cell. This is going to one daughter cell. Oops, daughter cell. And this is going to the other daughter cell. When the cell divides into two cells, each of the daughter cells will inherit one daughter DNA. So I'm just going to make up a DNA code. We have 3' ATGCCG. I'm just making something up. AGAGA. Okay. 5'. So the parent strand, this is the parent strand. So complementary base pairing rules. I'm not drawing it as a double helix because I'm not a good enough artist. But C's are always bound to G's and A's are always bound to T's. And we have the anti-parallel nature. See, 3' to 5', 3' to 5'. Each strand runs opposite of the other strand in terms of their sugar phosphate backbones. Now, to the first step of DNA synthesis, the two strands have to be separated. There's an enzyme that does this called DNA helicase. So I'm going to erase this parent strand and draw it farther apart, farther away. So now the two parent strands have been separated from each other. The next step is to build the complementary daughter strands. The daughter strands are going to be built by an enzyme called DNA polymerase. Now, anytime you see an ASE at the end of the name of a molecule, you can assume it's an enzyme that catalyzes chemical reactions. Okay? So DNA polymerase, remember a polymer, is a large molecule built out of repeating subunits strung together. So polymerase builds polymers. This is DNA polymerase, meaning that the polymer that this enzyme builds is DNA. So DNA polymerase builds DNA. So it's going to run along, and it always reads 3' to 5', and it builds 5' to 3'. So I'm going to draw the daughter strands in blue. The parent strands will be in red. So it's going to start here on this strand and build T, A, C, G, G, C. So remember, it's just complementary base pairing rules. It's just filling in, it's reading what the order of nucleotides is and adding the complementary nucleotides. The new strand is also anti-parallel to the original parent strand. Now, this strand at the bottom, it has to be read and built in this direction, because remember, it's being read 3' to 5' and built 5' to 3'. So it's anti-parallel, so the 5' end of the daughter matches with the 3' end of the parent. Complementary, so T's to A's, C's to G's, T's to A's, C's to G's, T's to A's, C's to G's, G's to C's, G's to C's, C's to G's, A's to T's, T's to A's, 3'. So they're built in different directions because this enzyme can only travel along the parent strand 3' to 5'. And so it must build 5' to 3'. And because the two parent strands were running in opposite directions from each other, because the parent strands were anti-parallel, they need to be read and built in opposite directions from each other. Okay, so now if you look at these two molecules, if you look at them, they are genetically identical to each other. This is clonal reproduction. If we're looking at the bottom strand, ATG, and at the top strand, ATG, right? At the bottom strand, CCG is next. CCG is next on the top strand. These two daughters are identical to each other. They're also identical to the original parent strand. Okay, so that is DNA replication, the basics of it. Now, there are some complicated features that we'll get into. One thing that's complicated is that this molecule is really, really long. It's, like I said, there's 6 feet in every single human cell. It would take forever. It would take much too long to just start at one end and finish at the other. So instead, you have many helicases and many DNA polymerases that attach at different points and all start at different points and replicate together. And I'll show you that in the next slides. Another complicating factor is that it's really just one big enzyme conglomerate that is replicating the two strands. You don't have 2 different enzymes attached to the 2 different parent strands. One enzyme is attached to the 2 parent strands, but it has to read each one in opposite directions, and that's a little bit complicated. So here you see a replication fork. You see that the parent strand is being unzipped, and where it's unzipping, that's the replication fork. And the replication fork moves as more of the molecule gets unzipped. And you can see that as it unzips, these daughter molecules are being built. So replication of a DNA molecule begins at particular sites called origins of replication. Now, origins of replication are short stretches of DNA that have a specific sequence of nucleotides, and they're repeated throughout the longer molecule. So, for example, let's just say we have this huge molecule of DNA. The origin of replication will have a specific sequence that's kind of long. But let's just, for argument's sake, just as the demonstration, say the origin of replication has the sequence CCACC. I'm just making something up. That's not really it. But I just want to demonstrate to you what this bullet point means. Every place that has a CCACC, that's going to be an origin of replication. So helicase is going to bind here, and another helicase will bind here and begin unzipping. Another helicase will bind here. And everywhere the helicase binds, it's going to begin unzipping the parent strand of DNA at these points, at these origins of replication. So proteins that initiate DNA replication, helicase and DNA polymerase, they're going to attach to the DNA at the origin of replication, and they're going to separate the 2 strands of the double helix. Replication then proceeds in both directions from the origin of replication, creating these replication bubbles. So let me show you again what I mean. So this is a double strand, okay? And of course, the complement to the origin of replication would be here. G, G, T, G, G. If we're going to do both strands, right? We've got to be accurate here. There's 2 strands. So you're going to end up with these growing bubbles, and we call them replication bubbles. The bubbles began at the origin of replication. At each origin of replication, right? So helicase, there's 2 helicases unzipping in each direction. These are the origins of replication. Helicase bound to the origin of replication and began unzipping the DNA in both directions outwards, creating these growing replication bubbles. So here's a prettier picture of that. These are the origins of replication in green. Helicase binds there, begins unzipping the DNA in both directions. DNA polymerase is going to follow. So as this is the replication fork, as helicase unzips it, DNA polymerase follows, building the daughter strands behind. This is the daughter strands in gray. And these replication bubbles grow and grow and grow, as you can see here. They're growing and growing until they merge with each other. And once they merge together, then you have 2 two separate yet identical daughter strands of DNA. This is much faster than just having one helicase and one DNA polymerase start at one end of the DNA molecule and travel linearly all the way through. Instead, you have many of them working in parallel. So they start at different points and they all move outwards towards each other until they meet up, and then the daughter strands are finished. So remember, helicase is the enzyme that unzips the parent DNA. DNA polymerase reads the parent strand and builds a complementary daughter strand. And then there's one more enzyme I want you to know, and that's ligase. Ligase fuses DNA fragments together. Now, one place that ligase is going to do that is to merge all these growing daughter strands together. So ligase has to fuse here. Ligase fuses the daughter strands there and there where these replication bubbles met up. You have to remember that DNA polymerase reads the parent strands in the 3' to 5' direction, starting at the 3' end, moving toward the 5' end. And the complementary daughter strands are anti-parallel, and so they are built in the 5' to 3' direction. Both strands are read and built at the same time by DNA polymerase. Now, this creates a problem for one of the strands because the two strands run in opposite directions from each other. So how can one enzyme read them both and build the complement at the same time? So helicase is opening up the parent strand in that direction, and DNA polymerase is following it, building the daughter strands. Now, the thing is, this parent strand runs 3'. 3' is here and 5' is here. And so here, this gray triangle thing, that's supposed to be DNA polymerase. It's reading the parent strand and synthesizing the daughter strand, the complement. And this one at the top can just go in a straight line. It's really simple for it. So this daughter strand is synthesized continuously. We call it the leading strand. Now, things aren't so simple for the strand down here. We call it the lagging strand because, see, the 3' end is on the wrong side and the 5' end is on the wrong side. So it needs to move in the opposite direction, but it needs to keep moving forward with helicase. Helicase is moving this way. So this, on the bottom, the DNA polymerase has to move in this image from right to left, yet it's only allowed to move left to right because left to right we have 3' to 5' for this strand. So what it does is it goes like this. It makes the lagging strand in segments. So it can only move 3' to 5'. So it moves this way and reads and synthesizes that way. Then it goes backwards and does the next segment, and then goes the next segment, then the next segment, then the next segment. So it is ultimately moving from the right to the left, but it's doing it kind of like, okay, and it like, so it has to keep going backwards and then catching up, and then going backwards and then catching up. So we call this the lagging strand. I know this is difficult to understand. We'll look at several images to try to parse it all out, okay? And then DNA ligase, there's that ligase. It comes along and fuses these segments together. The little fragments that the lagging strand is built out of are called Okazaki fragments. And ligase has to fuse the Okazaki fragments of the lagging strand together. The overall direction of replication is this way. Both strands are being read 3' to 5', and both strands are being built 5' to 3'. But the 3' and 5' direction are reversed in the two strands because they're anti-parallel. So this is 3' to 5' for this strand. This is 3' to 5' for this strand. The overall direction of replication has to be this way, so it has to be built like this. So this is 1, 2, 3, 4, and so on and so forth. So its overall direction is going to be right to left, even though it's made in segments left to right. So DNA polymerase can only travel the parent strand in the 3' to 5' direction. The leading strand is copied continuously, 3' to 5'. The lagging strand runs anti-parallel to the leading strand, so it cannot be copied continuously. DNA polymerase reaches ahead in the 3' direction and begins synthesizing the complementary daughter strand 3' to 5', but in the reverse spatial direction relative to the leading strand. I'm going to edit this. When the new segment meets up with the previously completed segment, DNA polymerase will reach ahead again and synthesize the next segment. So it just does it like over and over again. Reaches ahead, runs backwards. Reaches ahead, runs backwards. And that's the only way that it can run 3' to 5'. These segments are called Okazaki fragments, and they must be fused together by ligases. I'm linking to a video where you can better visualize the production of the leading strand and the lagging strand, how the Okazaki fragments are made and why. So just click on the link and you can see it in action. I think seeing it done in real time makes it much easier to understand. DNA polymerase has proofreading capabilities, which makes it very accurate. It only makes one mistake in a billion. DNA polymerase and DNA ligase also repair DNA that's been damaged by harmful radiation, toxic chemicals, things like that. So DNA replication ensures that all the somatic cells in a multicellular organism carry the same genetic information. The idea is to get the daughters to be identical to the parent. Otherwise, things would be such chaos. If we were experiencing constant mutations, most of which were negative, caused our cells to be dysfunctional, that would not be very good for survival. So mutations are very rare. Mutations are just mistakes that DNA polymerase makes when it's copying the genetic code. I'm linking to yet another video of DNA synthesis in real time so you can see the overall process from beginning to end. Now, before we end, I want to talk a little bit more about mutations. Now, there are many protective measures against them. The enzyme that copies DNA from parent to daughter cells, DNA polymerase, has the capacity to proofread and correct errors as it goes. Only 1 out of a billion nucleotides are mutated. Now, the average gene is 1,000 nucleotides long, so 1 out of every million genes is mutated. Now, how many genes does an organism have? Well, in a bacterial colony of 10 million cells, which is a very reasonable size for a bacterial colony, there will be several mutants in every generation. So let's take staph infection. Staph infection is caused by a bacteria called Staphylococcus aureus, and Staphylococcus aureus divides once every 30 minutes under ideal conditions, which means that its population doubles in number every 30 minutes. Now, the genome for Staphylococcus aureus is 2,800,000 nucleotides in length. Now, if you started with one bacterium, one Staphylococcus aureus, and it was in ideal conditions, so it was replicating at its ideal pace, doubling in number every 30 minutes, then after 30 hours, after a little more than a day, you would have, I don't even know what number this is, 1 times 10 to the 18th cells. That's 1 followed by 18 zeros. That's how many bacteria would be in that colony after just 30 hours. So to figure out how many nucleotides, you're trying to figure out how many mutants there are, right? And you know the mutation rate is 1 in a billion nucleotides. So how many nucleotides are in that whole colony at the end of 30 hours? You would have to multiply this number by this number to figure out how many nucleotides were in that colony of Staphylococcus aureus after 30 hours. I'm not going to bother performing that calculation because obviously it's huge. And it's huge enough that every possible mutation has occurred in this population 30 times. So, and that's just after a little more than a day. So mutations are very rare when you're looking at it from the nucleotide perspective. 1 in every billion nucleotides, 1 in every billion letters. That's very accurate. That's almost 0 mutations, but it's not 0 mutations. But so when you think about how huge a genome is, it takes a lot of nucleotides, it takes a lot of letters to instruct the build of any organism, even a bacterium. And then you think how many organisms are on the planet. There's so many organisms on the planet. There's so many bacteria in just one little bacterial colony. And so when you look at it from the perspective of how many mutants are there in a population, There's lots. The next video in this series will be a cursory overview of viruses and bacteria.